Hi class, this is your instructor, Skylar Huff. We're now in Chapter 15. This is genes and how they work. You've seen how genes specify traits and how those traits can be followed in genetic crosses. You've also seen that information in genes resides in the DNA model. Given that, the picture you see here shows all the DNA within the entire E. coli chromosome. Information in DNA is replicated by the cell and then partitioned equally during the process of cell division. The information in DNA is much like a blueprint of a building. The construction of the building uses the information in the blueprint, but requires building materials and carpenters and other skilled laborers using a variety of tools working together to actually construct the building. Similarly, the information in DNA requires nucleotide and amino acid building blocks, multiple forms of RNA, and many proteins acting in a coordinated fashion to make up the structure of a cell. We will now turn to the nature of genes themselves and how cells extract the information in DNA in the process of gene expression. Gene expression can be thought of as the conversion of genotype into phenotype. So throughout this chapter we'll go through roughly 8 sections, skipping that 15.3 section with just one thing mentioned there. We know that DNA encodes proteins, but this knowledge alone tells us little about how the information in DNA can control cellular functions. Researchers had evidence that genetic mutations affected proteins, and in particular enzymes, long before the structure and code of DNA was known. Here, we review the evidence of the link between genes and enzymes. In 1902, British physician Archibald Garrard noted that certain diseases among his patients seemed to be more prevalent in particular families. By examining several generations of these families, he found that some diseases behaved as though caused by simple recessive alleles. Garrard concluded that these disorders were Mendelian traits, and that they had resulted from changes in the hereditary information in an ancestor of the affected families. Garrard investigated the disorder alkaptonuria. So again, that's alkaptonuria. In detail. Parents with alkaptonuria produce urine-containing homogenistic acid alkapton, which is rapidly oxidized in air, turning the urine black. In normal individuals, homogenistic acid is broken down into simpler substances. Garrard concluded that patients with alkaptonuria lack the enzyme necessary to catalyze this breakdown, further speculating that many other inherited diseases might also reflect enzyme deficiencies. Garrard was actually ahead of his time, as the connection between Mendelian alleles and enzymes was not codified for almost 40 years. In 1941, the link between traits followed in crosses and proteins was demonstrated by a series of experiments by George Beadle and Edward Tatum at Stanford University. They used the then new technology of inducing mutations by X-rays to collect a series of mutants for detailed study. The work is summarized here. Beadle and Tatum chose the bread mold Neurospora crassa for their experiments. This fungus can be grown readily in the laboratory on a defined medium consisting of only a carbon source, glucose, and a vitamin, biotin, plus inorganic salts. This is called minimal medium because it represents the minimal requirements to supplement growth. Any cells that can grow on minimal medium must be able to synthesize all necessary biological molecules. When Beadle and Tatum exposed spores to X-rays, they expected to find some mutants that would be unable to grow on minimal medium. These so-called nutritional mutants result from damage to genes encoding functions necessary to make biological molecules. To identify nutritional mutants, Beadle and Tatum first grew cultures on rich medium, then placed subcultures of individual fungal cells onto minimal medium. This identifies any cells that had lost the ability to make compounds necessary for growth. They concentrated on the ability to synthesize the amino acid arginine, which the biosynthetic pathway was known. To identify mutants unable to make arginine, they selected cultures that would grow only on minimal medium plates plus arginine. This resulted in a collection of independent mutants that were all unable to synthesize arginine and that could be genetically mapped to different chromosomal locations. This defined 4 genes named ARG-E, ARG-F, ARG-G, and ARG-H, seen there at the right. With these tools in hand, Beadle and Tatum were able to genetically dissect the arginine biosynthetic pathway by supplementing minimal medium with intermediates in the biochemical pathway. They could identify the specific lesion in each mutant. For any indeterminate, if the mutation affects an enzyme that acts early in the pathway than the supplement, then growth should be supported, but not if the mutation affects a step after the intermediate used, and that's being shown to you all here. Using this approach, Beadle and Tatum were able to isolate a mutant strain defective for each enzyme in the biosynthetic pathway. Thus, each of the mutants they examined had a defect in a single enzyme, caused by a mutation at a single site on a chromosome. Beadle and Tatum concluded that genes specify the structure of enzymes, and that each gene encodes the structure of one enzyme, as you see here. So given this, they called this relationship one gene, one enzyme hypothesis. Today, because many enzymes contain multiple polypeptide subunits, each encoded by a separate gene, the relationship is generalized to the one gene, one polypeptide hypothesis. This hypothesis clearly states that the molecular relationship between genotype and phenotype. As you learn more about genomes and gene expression, you will see that this clear relationship is overly simplistic. Eukaryotic genes are more complex than those of prokaryotes, and some enzymes are composed, at least in part, of RNA, itself an intermediate in the production of proteins. Nevertheless, one gene, one polypeptide is a useful starting point for thinking about gene expression. The conversion of genotype to phenotype requires information stored in DNA to be converted to protein. The nature of this information flow in cells was first described by Francis Crick as the central dogma of molecular biology. This is important to know. Information passes in one direction from the gene, DNA, which is actually going to go through the process of replication. So it's DNA to an RNA copy of the gene, and the RNA copy directs the sequential assembly of the chain of amino acids into a protein. I'll show a photo. This is it. So stated briefly, DNA to RNA to protein is that central dogma of molecular biology. The central dogma provides an intellectual framework that describes the information flow in biological systems. We call the DNA to RNA step transcription because it produces an exact copy of the DNA. Much of the legal transcription contains the exact words of a court proceeding. The RNA to protein step is termed translation because it requires translating from the nucleic acid to the protein languages. Since the original formulation of the central dogma, a class of viruses called retroviruses was discovered that can convert their RNA genome into a DNA copy using the viral enzyme reverse transcriptase. This conversion violates the direction of informational flow of the central dogma and the discovery forced an updating of the possible flow of information to include this reverse flow from RNA to DNA. And given what's in your textbook, as far as we call DNA to RNA step transcription, I'll say because it produces ribonucleic acid. Because I would say replication produces an exact copy of the DNA. The process of transcription produces an RNA copy of the information in DNA. That's why I say this, because I think there is a mistake in this textbook. Hence I reiterated. We call the DNA to RNA step transcription because it produces an RNA copy of the information in DNA. That is, transcription is the DNA-directed synthesis of RNA by the enzyme RNA polymerase. This process uses the principle of complementarity, first described back in Chapter 14, to use DNA as a template for RNA. Because DNA is double-stranded and RNA is single-stranded, only one of the two DNA strands needs to be copied. We call the strand that is copied the template strand, shown here at the top of the screen. The RNA transcripts sequence is the complementary to the template strand. I repeat, the RNA's transcript sequence is complementary to the template strand. The strand of DNA not used as a template is called the coding strand. So you should know how to go through this based on what you're given, by way of that coding strand to the template strand, or sense to antisense, to the coding mRNA sequence. So the DNA strand that is not used as a template is called the coding strand. It has the same sequence as the RNA transcript, except that U for uracil in the RNA is the T for thymine in the DNA coding strand. So if, again, you can look at the screen right now. If you look at the coding strand, or that sense strand, instead of a thymine being first at that 5' end, we have a uracil. We still have cytosine, we still have adenine, we still have guanine, and I'll stop. But you do see yet again another thymine on that coding or sense strand, and you then also will see a uracil instead, and that is shown right here. Whoops, shown right here. Another naming convention for the two strands of the DNA is to call the coding strand the sense strand, as it has the same sense as the RNA. The template strand would then be the antisense strand. The RNA transcript used to direct the synthesis of polypeptides is termed messenger RNA, or mRNA. Its name reflects its use by the cell to carry the DNA message to the ribosome for processing. The process of translation is by necessity much more complex than transcription. In this case, RNA cannot be used as a direct template for a protein because there is no complementarity. That is, a sequence of amino acids cannot be aligned to an RNA template based on any kind of chemical fit. Molecular geneticists suggested that some kind of adaptive molecule must exist that can interact with both RNA and amino acids, and transfer RNA, or tRNA, was found to fill this role. This need for an intermediary adds a level of complexity to the process that is not seen in either DNA replication or transcription of RNA. Translation takes place on the ribosome, the cellular protein synthesis machinery. So again, I say, translation takes place on the ribosome, the cellular protein synthesis machinery, and it requires the participation of multiple kinds of RNA and many proteins. Here, it will be provided an outline of the processes, all are described in detail in the rest of this chapter. All RNAs are synthesized from a DNA template by transcription. Gene expression requires the participation of multiple kinds of RNA, each with different roles in the overall process. Here is a brief summary of these roles, which are described in detail in the remainder of this chapter. Messenger RNA. Even before the details of gene expression were unraveled, geneticists recognized that there must be an intermediate form of information in the DNA that can be transported out of the eukaryotic nucleus to the cytoplasm for ribosomal processing. This hypothesis was called the messenger hypothesis, and we retain this language in the name messenger RNA, or mRNA. Ribosomal RNA. This class of RNA is found in ribosomes and is called rRNA. There are multiple forms of rRNA, and rRNA is found in both ribosomal subunits. This rRNA is critical to the function of the ribosome. Transfer RNA. The intermediary adaptive molecule between mRNA and amino acids is tRNA. transfer RNA molecules have amino acids covalently attached to one end and an anticodon that can base pair with an mRNA codon at the other. The tRNAs act to interpret information in mRNA and to help position the amino acids on the ribosome. Small nuclear RNA, or snRNAs, they are a part of the machinery that is involved in the nuclear processing of the eukaryotic pre-mRNA, srpRNA. In eukaryotes, where some proteins are synthesized by ribosomes on the rough endoplasmic reticulum, this process is mediated by the signal recognition particle, or SRP. Small RNAs. This class of RNAs includes both microRNA, or miRNA, and small interfering RNA, or siRNA. These are involved in the control of gene expression, which I will not go into that. How does a sequence of nucleotides in a DNA molecule specify the sequence of amino acids in a polypeptide? The answer to this essential question came in 1961 through an experiment led by Francis Crick and Sidney Brenner. The experiment was so elegant and the result so critical to the understanding of the genetic code that we describe it here in detail. Crick and Brenner reasoned that the genetic code most likely consisted of a series of blocks of information called codons, each corresponding to an amino acid in the encoded protein. They further hypothesized that the information encoded within one codon was probably a sequence of 3 nucleotides. With 4 DNA nucleotides, guanine, cytosine, thymine, and adenine, using 2 in each codon can only produce 4 to the second power, or 4 squared, which is 16 different codons, which is not enough to code for 20 amino acids. However, 3 nucleotides results in 4 to the third power, or 4 to the third, or 64 different combinations of 3. More than enough. In theory, the sequence of codons in a gene could be punctuated with nucleotides between the codons that are not used, like the spaces that separate the words in this sentence. Alternatively, the codons could lie immediately adjacent to one another, forming a continuous sequence of nucleotides. If the information in the genetic message was separated by spaces, then altering any single word would not affect the entire sentence. In contrast, if all the words are run together but read in groups of 3, then any alteration that is not in groups of 3 would alter the entire sequence. These 2 ways of using information in DNA imply different methods of translating information into a protein. To choose between these alternative mechanisms, Crick and his colleagues used a chemical to create mutations that caused single base insertions or deletions from a viral DNA molecule. They then showed that combining an insertion with a deletion restored function, even though either one individually displayed loss of function. In this case, only the region between the insertion and the deletion would be altered. By choosing a region of the gene that encoded for a part of the protein not critical to function, this change did not cause a change in phenotype. When they combined a single deletion or 2 deletions near each other, the genetic message shifted, altering all of the amino acids after the deletion. When they made 3 deletions, however, the protein after the deletions was normal. They obtained the same results when they made additions to the DNA consisting of 1, 2, or 3 nucleotides. Thus, Crick and Brenner concluded that the genetic code is read in increments of 3 nucleotides. In other words, it is a triplet code, and that reading occurs continuously without punctuation between the 3 nucleotide units. And that is shown to you all here, the triplet code. These experiments indicate the importance of the reading frame for the genetic message. Because there is no punctuation, the reading frame established by the first codon in the sequence determines how all subsequent codons are read. We now call the kinds of mutations that Crick and Brenner used, frame shift mutations, because they alter the reading frame of the genetic message. So this is what Crick and Brenner did along with their colleagues. The determination of which of the 64 possible codons encoded each particular amino acid was one of the greatest triumphs of the 20th century biochemistry. Accomplishing this decryption depended on two related technologies. One, cell-free biochemical systems that will support protein synthesis from a defined RNA, and two, the ability to produce synthetic defined RNAs that could be used in the cell-free system. And though I showed this and didn't really do much with it, let me slow it down a bit. So this is getting to that triplet code. If you look closely, we have MET, which is methionine from that AUG or start codon. Then we have the next amino acid. I'm at the top with the one base deleted. This is, I'm not sure which amino acid is maybe proline, which comes from CCU. Threonine, which is ACG. Since that one base is deleted, instead of being ACG, it becomes AGC. That is a point mutation. One base deleted, and things have gotten shifted down from that 3' end to the left to that 5' end. All amino acids have now changed after that point mutation. Whereas at the bottom, the 3 bases deleted, or that frame shift mutation, you have that very same wild type, methionine, protein, threonine. So instead of ACG being that third codon, we now have CAC, and that is now, since that has occurred, the amino acids, it says, do not change after the third deletion. And they did not. They just shifted down from that 5' end. They shifted from that 3' end on the right down toward that 5' end on the left because the 3 bases were deleted. And that is that frame shift mutation, which is what allowed Crick and Brenner to make this discovery. So during a 5-year period from 1961 to 1966, work performed primarily in American biochemist Marshall Nirenberg's laboratory led to the elucidation of the genetic code. Nirenberg's group first showed that adding the synthetic RNA molecule, poly-U, an RNA molecule consisting of a string of uracil nucleotides, to their free cell system produced the polypeptide polyphenylalanine, which is a string of phenylalanine amino acids. Therefore, U encodes phenylalanine. Next, they used enzymes to produce RNA polymers with more than one nucleotide. These polymers allow them to infer the composition of many of the possible codons, but not the order of the bases in each codon. The researchers then were able to use enzymes to synthesize defined 3 base sequences that could be tested for binding to the protein synthesis machinery. This so-called triplet binding assay allowed them to identify 54 of the 64 possible triplets. The organic chemist H. Gobine Khorana provided the final piece of the puzzle by adding organic synthesis to produce artificial RNA molecules of defined sequence, and then examining what polypeptides they directed in cell-free environments. The combination of all of these methods allowed the determination of all 64 possible nucleotide sequences, and the full genetic code was determined, shown here. So I've provided this in the past for you all to go through the process, not merely from just DNA to RNA, but also from DNA to RNA, now to protein. That is what you all should be able to do. Some obvious features of the code jump out of the table, and that's referring here to table 15.1. First, 61 of the possible 64 codons are used to specify amino acids. 3 codons, UAA. And if you start here on that first letter side, go to the top second letter, A, and then go to third letter, A, UAA, UGA and UAG are reserved for another function. They signal stop and are known as stop codons. The only other form of punctuation in the code is that AUG is used to signal start and is therefore the start codon. And you see here A first letter, U second letter, third letter, G, AUG, methionine start. So this is where we are. So in this case, the codon has a dual function because it also encodes the amino acid methionine, that's MET, which was there for that start codon. You can see that 61 codons are more than enough to encode 20 amino acids. That leaves a lot of extra codons. One way to deal with this abundance would be to use only 20 of the 61 codons, but this is not what cells do. In reality, all 61 codons are used, making the code degenerate, which means that some amino acids are specified by more than one codon. The reverse, however, in which a single codon would specify more than one amino acid, is never found. This degeneracy is not uniform. Some amino acids have only one codon, and some have up to 6. In addition, the degenerate base usually occurs in position 3 of a codon, which I have sometimes said to be on that 3' end, such as the first two positions are the same, and 2 or 4 of the possible nucleotides at position 3 encode for that same amino acid. The nature of protein synthesis on ribosomes explains how this codon usage works and is discussed in section 15.7. The genetic code is the same in almost all organisms, so this is why you may have heard me say the genetic code is, well, practically universal. So the universality of the genetic code is among the strongest evidence that all living things share a common evolutionary heritage. Because the code is universal, genes can be transferred from one organism to another and can be successively expressed in their new host, as you now see here. The universality of gene expression is central to many of the advances of genetic engineering discussed in Chapter 17, which is the next chapter. In 1979, investigators began to determine the complete nucleotide sequences of the mitochondrial genomes in humans, cattle, and mice. It came as something of a shock when these investigators learned that the genetic code used by these mammalian mitochondria was not quite the same as the universal code that had become so familiar to biologists. In the mitochondrial genomes, what should have been a stop codon, UGA, was instead read as the amino acid tryptophan. AUA was read as methionine rather than isoleucine, and AGA and AGG were read as stop codons rather than arginine. Furthermore, minor differences from the universal code have been found in the genomes of chloroplasts and in ciliates, which are certain types of protists. Thus, it appears that the genetic code is not quite universal. Some time ago, presumably after they began their endosymbiotic existence, mitochondria and chloroplasts began to read the code differently, particularly the portion associated with the stop signals. And as I mentioned, shown to you here, the piglet on the right is a conventional piglet. The piglet on the left was engineered to express a gene from jellyfish that encodes for green fluorescent protein. The code on the piglet's nose is due to the expression of this introduced gene. Such transgenic animals indicate the universal nature of the genetic code. So that the one thing I'll mention from that 15.3 section is before you here. In prokaryotes, the mRNA produced by transcription begins to be translated before transcription is finished. That is, transcription and translation are coupled. As soon as that 5' end of the mRNA becomes available, ribosomes are loaded onto this to begin translation. In eukaryotes, where transcription occurs in the nucleus and is followed by translation in the cytoplasm, this kind of coupling does not occur. So if you did not hear that, In prokaryotes, there are no necessary differences in site of process. Replication, transcription, and translation, they all occur there in the cytoplasm in prokaryotes, whereas in eukaryotes, the locations are different. Another difference between prokaryotic and eukaryotic gene expression is that mRNA produced in prokaryotes may contain multiple genes. Prokaryotic genes are often organized such that genes coding related functions are closer together. This grouping of functionality related to genes is referred to as an operon. An operon is a single transcription unit that encodes for multiple enzymes necessary for a biochemical pathway. So we will not get to that in this course, though if you are interested, I can certainly provide you more information. So this speaks to those operons here very briefly. So an operon, single mRNA containing multiple genes, such as the LAC operon or the TRIP operon. The basic mechanism of transcription by RNA polymerase is similar in all systems. In fact, RNA polymerases of bacteria and archaea and eukaryotes all appear to be descended from the same ancestral enzyme. However, the details of RNA production in eukaryotes differ enough to merit a bit of consideration. An obvious difference between bacteria and eukaryotes is that bacteria have a single RNA polymerase, while eukaryotes have 3 distinct RNA polymerases with different cellular roles. The enzyme RNA polymerase 1 transcribes rRNA, RNA polymerase 2 transcribes mRNA, and some small nuclear RNAs, and RNA polymerase 3 transcribes tRNA and some other small RNAs. Together, these 3 enzymes accomplish all transcription in eukaryotic cells. The 3 eukaryotic RNA polymerases require different control elements in the DNA to allow each polymerase to recognize where to initiate transcription. Each polymerase recognizes a different promoter structure, which I will not get too specific on the different promoter structures. I will not. So RNA polymerase 1 promoters, those promoters are species-specific. RNA polymerase 2 promoters, they consist of that core promoter that can be composed of a number of elements, which includes the TATA box, and the TATA sequence is upstream of the start site, i.e. it's before it. And then RNA polymerase 3 promoters, I said then RNA polymerase 3 promoters, most are found within the gene itself. So as far as the initiation of transcription at polymerase 2 promoters, so it requires a series of transcription factors. So transcription factors, they're there and they're necessary to get the RNA polymerase 2 enzyme to a promoter and to initiate gene expression. They interact with RNA polymerase to form the initiation complex at that promoter sequence or at the promoter. So elongation complex factors coordinated by structural feature called carboxyl terminal domain. And then additional factors allow RNA polymerase to transcribe DNA that is assembled as chromatin, including FACT or facilitates chromatin transcription and chromatin remodeling complexes. So I say all that to put it this way. The initiation at RNA polymerase 2 promoters is analogous to what you didn't get a moment ago with the prokaryotes. Though what's unique is, instead of a single factor allowing promoter sequence recognition, eukaryotes use a host of transcription factors, and these factors cooperatively act in recruitment of RNA polymerase 2. I repeat, these factors act cooperatively to recruit RNA polymerase 2 to a promoter. And then I said the transcription factors, they interact with RNA polymerase 2 to form an initiation complex at the promoter sequence, which let's just show that now. So we'll explore this now and that'll be it. So number 1, the transcription factor recognizes and binds to the TATA box sequence, which is part of that core promoter region. The other transcription factors are then recruited and the initiation complex begins to build. And then lastly, RNA polymerase II associates with the transcription factors and the DNA, forming the initiation complex, and transcription will now begin, as you see there at number 3. Let's modify things here. A distinct feature of eukaryotic transcription is at the 3' end of mRNA and is not the end of the transcript. I repeat, this distinctive feature of eukaryotic transcription that is at the 3' end of the mRNA is not the end of the transcript. The transcript is cleaved downstream of a specific site. It says AAUAAA, while the polymerase is elongating and a series of 100 to 200 adenosine residues. So it's the 3' poly A tail is added to the mRNA. And it's referred to here, at least if you have ever wondered, as being polyadenylation. The enzyme responsible for this is poly A polymerase. A complex of factors, including poly A polymerase, associates with the elongating poly A polyisomerase via polyphosphorylated CTD. That's carboxyl terminal domain. The actual mechanism of transcription termination is not clear, but it involves the polymerase pausing around the poly site, and while the free mRNA is being polyadenylated, the remaining transcript is progressively removed by an exonuclease and the elongation complex dissociated. Termination of transcripts that are not polyadenylated uses a mechanism that requires other factors. The one common feature to transcription termination in all systems is something that causes RNA polymerase to pause so that the elongation complex can be disassembled. So one more thing I'll mention is when the transcript reaches about 20 nucleotides, it is modified by the addition of GTP to the 5' phosphate group, forming what is known as the 5' cap. This cap is joined to the transcript by the 5' end, the only 5' to 5' bond found in the nucleic acids. So it's often referred to as a methyl G cap, and the structure is important for translation, RNA stability, and further processes. So we now have them both. And if you're wondering what both are, I'm referring to the addition of that 5' cap, the addition of that poly A tail, and the last portion of this will be the removal of those introns, the non-coding regions. So there you have it, 5' cap and poly A tail at that 3' end. It is clear from the preceding discussion that mRNA in eukaryotes is not the same as the primary transcript of a gene. With the modified 5' end, 5' cap, and a 3' end poly A tail produced by another enzyme, and the removal of introns, the mRNA is an extensively modified form of the primary transcript. The greatest modification to the primary transcript is the removal of non-coding sequences, which allows greater flexibility for eukaryotic gene expression. This process of pre-mRNA splicing is accomplished by a macromolecular machine called the spliceosome. Let's get to this complex topic and some of its implications. The first genes associated were prokaryotic genes found in E. coli and its viruses. A clear picture of the nature of gene expression emerged from these systems before any eukaryotic genes were isolated. It was assumed that all details would differ, and the outline of gene expression in eukaryotes would be similar. As we have seen, this proved to be wrong. Many eukaryotic genes were found to contain sequences that were not represented in the mRNA. It is hard to exaggerate how unexpected this finding was. A basic tenet of molecular biology based on E. coli was that a gene was collinear with its protein product. That is, the sequence of bases in the gene corresponds to the sequence of bases in the mRNA, which in turn corresponds to the sequence of amino acids in the protein. In the case of eukaryotes, genes can be interrupted by sequences that are not represented in the mRNA and the protein. The term split genes was used at that time, but now the nomenclature that has stuck describes the unexpected nature of these sequences. We call the non-coding DNA that interrupts the gene intervening sequences, or introns, and we call the coding sequences exons because they are expressed. So let's get to introns versus exons, as you see here now. It is still true that mature eukaryotic mRNA is collinear with its protein product, but a gene that contains introns is not. Imagine looking at an interstate highway from a satellite, scattered randomly along the bed of concrete would be cars, some moving in clusters, others individually. Most of the road will be bare. That is what a eukaryotic gene is like. Scattered exons embedded with much longer sequences of introns. I'll go back for just a moment. In humans, only 1 to 1.5% of the genome is devoted to exons that encode for proteins. Yes, only 1 to 2% of your entire genome codes for proteins. 24% is devoted to the non-coding introns within which these exons are embedded, which is kind of shown to you all here. You have your DNA template, you have your primary RNA transcript, and then you have your mature or secondary mRNA as it was once described. Let's get in here. The obvious question is, how do eukaryotic cells deal with the non-coding introns? The answer is that the primary transcript is cut and put back together to produce the mature mRNA. The latter process is referred to as pre-mRNA splicing, and it occurs in the nucleus prior to the export of the mRNA to the cytoplasm by way of nuclear pores. Let me stop. So the intron-exon junctions are recognized by small nuclear ribonucleoprotein particles called SNRPs or SNRPs. So what can I say? These intron-exon junctions are recognized by small nuclear ribonucleoprotein particles called SNRPs or SNRPs, which you see now here. The SNRPs. The SNRPs, or SNRPs, I don't know which one you all prefer, are complexes composed of snRNA and protein. So the SNRPs are then clustered together with other associated proteins to form a larger complex called the spliceosome, which is responsible for the splicing or removal of the introns. There you have it. For splicing to occur accurately, the spliceosome must be able to recognize the intron-exon junctions. Introns all begin with the same 2-base sequence and end with another 2-base sequence that tags them for removal. In addition, within the intron, there is a conserved A nucleotide called the branch point, which is important for the splicing reaction. The splicing process begins with the cleavage of the 5' end of the intron. This 5' end becomes attached to the 2' OH of the branch point A, forming a branch structure called the lariat, due to its resemblance to a cowboy's lariat in a rope. The 3' end of the first exon is then used to displace the 3' of the intron, joining the two exons together and releasing the intron as a lariat. The splicing process takes place during transcription. The transcript exits the enzyme near the carboxyl terminal domain, or the CDT, allowing recruitment of splicing factors and the spliceosomal RMPs. There is evidence that the rate of transcription which is not constant, can affect the splice site selection. Pausing of the enzyme and the formation of secondary structure in the transcript can make splice sites inaccessible. No rules govern the number of introns per gene or the sizes of introns and exons. Some genes have no introns, others have as many as 50. The sizes of exons range from a few nucleotides to 7,500 nucleotides, and the sizes of introns are equally variable. The presence of introns explains why so little of a eukaryotic genome is actually composed of coding sequences. And we'll get to this in Chapter 18. One explanation for the existence of introns suggests that the exons represent functional domains of proteins, and the intron-exon arrangements found in genes represent the shuffling of these functional units over long periods of evolutionary time. This hypothesis, called exon shuffling, was first proposed soon after the discovery of introns and has since been the subject of much debate over the years. The recent flood of genomic data has shed light on this issue by allowing statistical analysis of the placement of introns and on intron-exon structure. This analysis has provided support for the exon shuffling hypothesis for many genes. However, it is also clearly not universal because all proteins do not show this kind of pattern. It is possible that introns do not have a single origin and therefore cannot be explained by a single hypothesis. We call all of the RNAs produced from a genome the transcriptome, and all the proteins produced from a genome the proteome. If every gene was spliced to include all exons, then the number of genes and transcripts and proteins would be equal. Unfortunately, it is not so simple. A single primary transcript can be spliced into different mRNAs by using a different set of exons, a process called alternative splicing. Before the Human Genome Project, it was estimated that humans had 100,000 genes. The current number is down to 20,000 genes that encode for proteins. But more than 80,000 protein encoding transcripts have been identified. Alternative splicing provides a way out of this conundrum. High-throughput systems to sequence cellular RNAs have produced a flood of data on patterns of transcription. More than 200,000 transcripts have been detected, leading to estimates of up to 95% of human genes producing multiple splice products. Estimates of the average number of transcripts per gene vary widely from as many as 10 to as few as 1.5 per gene. A conservative estimate from Genome Database provides an estimate of 85,000 possible protein encoding transcripts. Unfortunately, only a minority of these are experimentally verified. The situation is further clouded by the observation that as many as 80% of protein encoding genes appear to have one predominant transcript. The predominant transcript and minor transcripts can switch with different cell and tissue types, while the overall level of transcription remains the same. While there are well-characterized examples of alternative splicing producing different functional proteins, the overall biological significance remains unclear. The importance of a normal splicing pattern is also suggested by the observation that human genetic disorders can be due to aberrant splicing. It is clear that the transcriptome is much more diverse than one gene equals one transcript, but it's not clear the extent to which alternative splicing contributes to proteome diversity. The actual size of the proteome is also not yet clear, although progress is being made using mass spectrometry. The ribosome is the key macromolecular machine involved in translation, but it requires the participation of mRNA, tRNA, and a host of other factors. Critical to this process is the interaction of ribosomes with tRNA and mRNA. To understand this, we first examine the structure of the tRNA adapted molecule and the ribosome itself. Each amino acid must be attached to a tRNA with the correct anticodon for protein synthesis to proceed. This covalent attachment is accomplished by the action of activating enzymes called aminoacyl tRNA synthetases. One of these enzymes is present for each of the 20 common amino acids. Transfer RNA is a bifunctional molecule that must be able to interact with mRNA and with amino acids. The structure of tRNAs is highly conserved in all living systems. It can be formed into a cloverleaf type of structure based on intramolecular base pairing that produces double-stranded regions. This primary structure is then folded in space to form an L-shaped molecule that has two functional ends, acceptor stem and anticodon loop. The acceptor stem is the 3' end of the molecule, which always ends in 5' CCA 3'. The amino acid is attached to this end of the molecule. The anticodon loop is the bottom loop of the cloverleaf, and it can base pair with codons mRNA. So base pairs with codons in mRNA. So you can now see the anticodon loop, and there you have it. The reaction catalyzed by the enzymes is called the tRNA charging reaction, and the product is an amino acid joined to a tRNA, now called a charged tRNA. An ATP molecule provides the energy for this endergonic reaction. The charged tRNA produced by the reaction is an activated intermediate that can undergo the peptide bond forming reaction without an additional energy input. The charging reaction joins the acceptor stem of the carboxyl terminus of an amino acid. Keeping this directionality in mind is critical to understanding the function of the ribosome. And this is because each peptide bond will be formed between the amino group of one amino acid and the carboxyl group of another amino acid. Let's get down into ribosomes. The synthesis of any biopolymer can be broken down into initiation, elongation, and termination. You have seen this division for DNA replication as well as for transcription. In the case of translation or protein synthesis, all three of these steps take place on the ribosome, the large macromolecular assembly consisting of RNA and proteins. Details of the process by which the two ribosome subunits are assembled during initiation are described shortly. For the ribosome to function, it must be able to bind to at least two charged tRNAs at once so that the peptide bond can be formed between their amino acids. So let's continue on. The bacterial ribosome contains 3 binding sites, and those will be summarized in your text. The A site, or aminoacyl, binds to the tRNA carrying the next amino acid to be added. The P site, peptidyl, binds to the tRNA attached to the growing peptide chain, and the E site, exit, binds to the tRNA that carried the previous amino acid to be added. Transfer RNAs move through these sites successively during the process of elongation. Relative to the mRNA, the sites are arranged 5' to 3' in the order of E, P, A. The incoming charged tRNAs enter the ribosome at the A site. Here's your visual. Transit through the P site and then leave via the E site, 5' to 3', along with the mRNA there attached. The two functions of the ribosome involve decoding the transcribed message and forming peptide bonds. The decoding function resides primarily in the small subunit of the ribosome. The formation of peptide bonds requires the enzyme peptidyltransferase, which resides in the large subunit. Our view of the ribosome has changed dramatically over time. Initially, molecular biologists assumed that the proteins in the ribosome carried out its function, and the tRNA was a structure scaffold necessary to hold proteins in the correct position. Now, this view has mostly been reversed. The ribosome is seen instead as rRNAs that are held in place by proteins. The faces of the two subunits that interact with each other are lined up with rRNA, and parts of both subunits interact with mRNA. tRNA and amino acids are also primarily rRNA. It is now thought that peptidyltransferase activity resides in rRNA in the large subunit. Let's get into the translation, you all. So as mentioned in section 15.2, the start codon is AUG, which also encodes the amino acid methionine. So the ribosome usually uses the first AUG it encounters in an mRNA strand to signal the start of translation. So the process of translation is one of the most complex and energy-expensive tasks that cells perform. An overview of the process is perhaps deceptively simple. The mRNA is started through the ribosome, while tRNAs carrying amino acids bind to the ribosome, where they interact with mRNA by base pairing with the mRNA's codons. The ribosome and tRNAs position the amino acids such that peptide bonds can form between each new amino acid and the growing polypeptide. In eukaryotes, initiation is similar to that in prokaryotes, although it differs in two important ways. First, in eukaryotes, the initiating amino acid is methionine rather than in formal methionine. Second, the initiation complex is far more complicated than in prokaryotes, which we did not review or go over. We're beginning and ending here with eukaryote translation by way of initiation coming first. So it contains 9 or more protein factors, meaning initiation here in eukaryotes does, many consisting of several subunits. Eukaryotic mRNAs also lack the ribosome binding sequence, so there is nothing that you all need to study and go back and find in your text. The small subunit binds to the mRNA initially by binding to the 5' cap of the mRNA. When the entire ribosome is assembled around the initiator, tRNA, and mRNA, the second charged tRNA can be brought to the ribosome to bind to the empty A site. This requires an elongation factor called EFTU, which binds to the charged tRNA and to the GTP. The peptide bond can then form between the amino acid of the initiator tRNA and the newly arrived charged tRNA at the A site or in the A site. The geometry of this bond is relative to the two charged tRNAs and is critical to understand the process. Remember that an amino acid is attached to a tRNA by its carboxyl terminus. The peptide bond is formed between the amino acid of the incoming amino acid in the A site and the carboxyl end of the growing chain in the P site. You all can now see here. The addition of successive amino acids in a series of events occur in a cyclic fashion, matching tRNA anticodon with mRNA codon. That's the first step of the cycle, and let's go to that now. So number 1 is located in the center of this cyclic photo, or this donut resembling photo. Number 1 in the dark blue to your left, matching tRNA anticodon with the mRNA codon. Each new charged tRNA comes to the ribosome bound to EFTU and GTP. The charged tRNA binds to the A site if its anticodon is complementary to the mRNA codon at the A site. After binding, GTP is hydrolyzed, and EFTU GTP dissociates from the ribosome to be recycled by another factor. This two-step binding and hydrolysis of GTP is thought to increase the accuracy of translation. 2. Peptide bond formation. Peptidyltransferase, located in the large subunit, catalyzes the formation of a peptide bond between the amino group of the amino acid in the A site and the carboxyl group of the growing chain. This also breaks the bond between the growing chain and the tRNA in the P site, leaving it empty, no longer charged. The overall result of this is to transfer the growing chain to the tRNA in the A site. Thirdly, translocation of the ribosome, shown there at the bottom right, in the kind of darker blue. We were just at light blue at number 2. After the peptide bond has been formed, the ribosome moves relative to the mRNA and the tRNAs. The next codon in the mRNA shifts into the A site, and the tRNA with the growing chain moves to the P site. The uncharged tRNA formerly in the P site is now in the E site, and it will be ejected in the next cycle. E for ejected. Let me stop. This translocation step requires the accessory factor EFG and the hydrolysis of another GTP. This elongation cycle continues with each new amino acid added. The ribosome moves down the mRNA in a 5' to 3' direction, reading successive codons, codon by codon by codon, you all. The tRNAs move through the ribosome in the opposite direction, from the A site to the P site, and finally to the E site before they are ejected as empty tRNAs, which can be charged with another amino acid and then used again and again and again. As earlier mentioned, there are fewer tRNAs than codons. This situation is easily rationalized because the pairing between the 3' base of the codon and the 5' base of the anticodon is less stringent than normal. In some tRNAs, the presence of modified bases with less accurate pairing in the 5' position of the anticodon enhances this flexibility. This effect is referred to as wobble pairing because these tRNAs can wobble a bit on the mRNA so that a single tRNA can read more than one codon in the mRNA. Elongation continues in this fashion until a chain terminating stop codon is reached. For example, UAA. These stop codons do not bind to tRNA. Instead, they are recognized by release factors, proteins that release the newly made polypeptide from the ribosome. And there you have it. Termination. In eukaryotes, translation can occur either in the cytoplasm or on the rough endoplasmic reticulum. Proteins that are translated on the rough endoplasmic reticulum are targeted there based on their own initial amino acid sequence. These ribosomes found on the rough endoplasmic reticulum are actively translating and not permanently bound to the endoplasmic reticulum. A polypeptide that starts with a short series of amino acids called a signal sequence is specifically recognized and bound by a cytoplasmic complex of proteins called the signal recognition particle, or SRP. The complex of signal sequence and SRP is in turn recognized by a receptor protein in the endoplasmic reticulum membrane. The binding of the endoplasmic reticulum receptor to this signal sequence SRP complex holds the ribosome engaged in translation of the protein on the endoplasmic reticulum membrane, a process called docking, which docking is shown to you here. As the protein is assembled, it passes through a channel formed by the docking complex into the interior ER compartment, the cisternal space. This is the basis for the docking metaphor. The ribosome is not actually bound to the endoplasmic reticulum itself, but with the newly synthesized protein entering the endoplasmic reticulum. The ribosome is like a boat tied to a dock with a rope. The basic mechanism of protein translocation across membranes by the signal recognition particle and its receptor and channel complex has been conserved across all three cell types: eukaryotes, bacteria, and archaea. Given that only eukaryotic cells have an endomembrane system, this universality may seem curious. However, bacteria and archaea both export proteins to their plasma membrane, and the mechanism used is similar to the way in which eukaryotes move proteins into the cisternal space in the endoplasmic reticulum. Once within the ER cisternal space or lumen, the newly synthesized protein can be modified by the addition of sugars, called glycosylation, and transported by vesicles on the Golgi apparatus. This is the beginning of protein trafficking pathway that can lead to other intercellular targets to incorporation into the plasma membrane or to release outside of the cell itself. In summary, because of the complexity of the process of gene expression, it is worth stepping back to summarize some key points. So let's get ready for that test here. The process of gene expression converts information in the genotype into the phenotype. A copy of the gene in the form of mRNA is produced by transcription, and the mRNA is used to direct the synthesis of a protein by translation. Both transcription and translation can be broken down into initiation, an elongation cycle, and termination. Those are processes that produce their respective polymers, and the same is true for DNA replication. Eukaryotic gene expression is more complex than that of prokaryotes, which I did not go over that for prokaryotes. The structure of eukaryotic genes with interrupted coding sequences complicates both the processes of gene expression and the nature of genetic information, which means that processing must occur between transcription and translation, and that one gene can produce multiple messages. Transcription in eukaryotes also takes place in the nucleus, whereas translation takes place in the cytoplasm. This necessitates that the mRNA must be transported through nuclear pores to the cytoplasm prior to translation, which I mentioned that a little earlier in this lecture. The entire eukaryotic process is summarized as I show you now the figure, and the differences in gene expression between prokaryotes and eukaryotes is summarized in table 15.2. But for the purposes of this course, I say please focus on the first column called characteristic and that third column called eukaryotes. The term mutation is defined as a heritable change to the genetic material. Traditionally, this was detected by a change in phenotype, but now in the age of genome sequences, mutations can be detected in the DNA. Changing a single base in a gene can have profound consequences, for example in sickle cell anemia. Is that shown here? It is. So a change of A to T in the beta globin gene results in glutamic acid being produced by valine. I repeat, the change of an A to a T by beta globin gene results in a glutamic acid being replaced by valine. Because valine is nonpolar, this causes the beta chains to aggregate into polymers, altering the shape of the cells, ultimately producing the clinical syndrome. And this is what's shown here. So if you look closely there, instead of glutamic acid, we now have valine, which leads to the tetramers forming those long chains when deoxygenated, distorting that normal red blood cell, which is a biconcave cell, to that sickle-shaped cell. Genetic differences between individuals in a population are called polymorphisms, which arise by the process of mutation. We saw in Chapter 13 that single nucleotide polymorphisms are useful in mapping genes. In this section, we'll consider the range of changes that can occur in the genome, and then in Chapter 18, we will look at the range of variation that this has produced in human populations. A mutation that alters a single base is termed a point mutation, which leads to a single nucleotide variation in populations. Let's get to three types. So as these polymorphisms or in populations, they are called single nucleotide polymorphisms, SNPs. The replacement of one base for another is called a base substitution mutation, which is now what you see here at the top. Base substitutions fall into two categories based on whether the new base is similar to the old one. If the change replaces a purine with another purine, for instance, A2G, or a purine with another or a pyrimidine to another pyrimidine, for example, a C to a T, we call this transition. If the change replaces a purine with a pyrimidine or the reverse, for example, a G to a C, we call this a transversion. Missense mutations. Because the genetic code is degenerate, a base substitution mutation may not change the encoded amino acid. If it does not, we say the mutation is synonymous or silent. If it changes an amino acid, we say the mutation is non-synonymous. We refer to non-synonymous changes as missense mutations, as the sense of the codon involved has been changed. We further classify non-synonymous changes as conservative or non-conservative. Conservative changes replace an amino acid with a chemically similar one, nonpolar for nonpolar, while non-conservative changes replace an amino acid with a chemically different one, charged for uncharged, for example. Nonsense mutations. So a mutation that changes a codon from one that encodes an amino acid to a stop codon is called a nonsense mutation. If you look at the bottom at D as in dog, that is a nonsense mutation. The stop codon will cause translation to terminate, prematurely producing a truncated protein. The length of the resulting protein depends on the location of the new stop codon in the gene. And as I go back to B as in boy, that is a silent point mutation because coding and mRNA, if you look at those, the amino acid has not changed. This missense, however, at C, if you look at the wild type at A, instead of it being arginine after tyrosine, it is now histine. I think that's histine, but HIS. So that is a definite missense. The gain of loss of 1 to 50 base pairs is called an indel for insertion, deletion. Since codons consist of 3 bases, insertions or deletions that do not occur in multiples of 3 will shift the reading frame in the mRNA. Such frame shift mutation randomizes the downstream sequence of amino acids. The consequences for a protein function depend on the location of the frame shift, but they usually inactivate the protein. Frame shift mutations often lead to premature termination, as 3 in 64 codons are stop codons, or roughly a stop every 20 amino acids in a random sequence. In section 50.2, we saw how Crick and Brenner used frame shift mutations to infer the triplet nature of the genetic code. A special case of insertions is a class of mutations first discovered in Huntington disease. The gene involved contains a sequence of 3 bases that are repeated, called a trinucleotide repeat or triple repeat. This repeat sequence is expanded in the disease allele relative to the normal allele. This kind of triplet repeat expansion has been found in other neurodegenerative disorders, and repeat expansion disorders share some unusual features of inheritance. The prevalence of this kind of mutation is unclear, but at present they have only been observed in humans and mice, implying that they may be limited to vertebrates or even mammals. Triplet repeat expansion can occur in the coding region or in the non-coding transcribed DNA. In the case of Huntington disease, the repeat unit is in the coding region of the gene, and the triplet encodes glutamine, and expansion results in polyglutamine region in the protein. A number of other neurodegenerative disorders are due to this kind of mutation. Point mutations affect a single site on a chromosome, but more extensive changes that can affect more than 50 base pairs are called structural variation. Many human cancers are associated with chromosomal abnormalities, so these can be clinically relevant. When genomic rearrangements result in differences in the number of copies of a particular genomic region, it is called copy number variation. This can be the result of loss of DNA by deletion or gain of DNA by duplication or insertion, shown here. A special case of possible copy number variation is the insertion of mobile genetic elements or mobile element insertion. The amount of DNA that can be deleted without drastic consequences is dependent on the region of the genome, but large deletions are usually lethal. One human syndrome caused by a deletion is cri de chat, which is French for cry of the cat, after the noise made by children with this syndrome. Cri de chat syndrome is caused by a large deletion of the short arm of chromosome 5. It usually results in early death, although many affected individuals show a normal lifespan. It has a variety of effects, including respiratory problems. The duplication of a chromosomal region may or may not lead to phenotypic consequences. Effects depend on the location of the breakpoints where the duplication occurred. If the duplicated regions do not lie within a gene, there may be no effect, i.e. a de novo mutation. If the duplication occurs next to the original region, it is termed a tandem duplication. Some gene families, such as the globin genes that encode for hemoglobin, arose as a result of tandem duplication. Material in a chromosome can become inverted or transferred between chromosomes, events called inversions or reciprocal translocations, respectively. In each case, the total amount of genetic material remains the same, but it has been rearranged. More complex rearrangements can also occur, but they are even more rare. An inversion that results in a segment of a chromosome is broken in two places, reversed or put back together. An inversion may not have an effect on phenotype if the sites where the inversion occurs do not break within a gene. In fact, although humans all have the same genome, the order of genes in all individuals in a population is not precisely the same due to inversions that occur in different lineages. If one chromosome is broken off and joined to another chromosome, we call this a translocation. Translocations are complex because they cause problems during meiosis when two different chromosomes try to pair with each other during meiosis, which can lead to unbalanced translocation chromosomes. Translocations can also move genes from one chromosome region to another in a manner that changes the expression of genes in the involved region. Two forms of leukemia have been shown to be associated with translocations that move oncogenes into regions of a chromosome where they are expressed inappropriately in blood cells. The gist of this is here. Mutations are the starting point of evolution. And as I said that they are the starting point of evolution, without a continuous source of new variation, evolution will not occur. However, mutations are much more often detrimental than beneficial to an individual. Thus, a delicate balance must exist between the amount of new variation that arises in a species and the health of individuals in the species. The larger scale alteration of chromosomes has also been important in the evolution, although its role is poorly understood. It is clear that gene families arise by the duplication of ancestral gene, followed by the functional convergence of the duplicated copies. It is also clear that even among closely related species, the number and arrangement of genes on chromosomes can differ. There have even been whole genome duplications in lineages. And lastly, about 80% of new germline point mutations arise in the paternal genome, and this rate also increases with paternal age. And I'm saying this because the mutation rate in humans was first estimated by J.B.S. Haldane, who used the prevalence of hemophilia in a population. Whole genome sequencing has now allowed direct measurement of mutation rate by sequencing large numbers of parent-offspring trios, so mom, dad, and child. So multiple studies of this sort estimate a mutation rate of 1.1 to 1.8 times 10 to the negative 8 per nucleotide per generation. In other words, this represents 44 to 82 new single individual V mutations. And given that, meaning it's just one, and that's a single nucleotide variation. So that's 44 to 82. With 1 to 2 of these affecting coding sequences. So much like the prevalence of aneuploidy in the maternal germline, this is probably a reflection of reproductive biology. The stem cell population that gives rise to sperm accumulates mutations with age. So given all of this, indels occur at a rate of about 1/5 to 1/10, and much larger lesions or duplications that causes the copy number variation occur at even lower rates. So this is estimated to be about one new large one per 42 births. So given all of this, the rate for mobile element insertion appears to be on the order of maybe 1 per 20 births. So these data will continue to shed light on human evolution, especially combined with the analysis of other primate species. And it is also relevant to human genetic diseases, as it now appears that de novo mutations may play a larger role than previously thought. So this has been your instructor, Skylar Huff.